Hi, everyone. Sirani Fernando here on behalf of Endpoints News, and thanks for joining us today for this webinar session titled Building Better Biologics, Overcoming Hurdles in Protein Therapy Manufacturing. Firstly, I'd like to thank Catalent for sponsoring this session, and I'm super pleased to be your moderator. In today's discussion, we'll explore key challenges and solutions around protein therapy manufacturing, touching on early development, tech transfer and scalability, AI and digital tools in manufacturing, regulatory considerations, and working with CDMO partners. We have a fantastic panel of industry experts joining us today, each bringing unique insights across biotech, big pharma, and manufacturing strategy. But before we meet them, I have a few housekeeping items to share with our audience. There will be time at the end of the discussion for us to answer some audience questions, so please hit the Q&A button at the bottom of your screen as soon as you think of a question. Additionally, this session is being recorded and will be available on demand tomorrow to rewatch or to share with colleagues. So to start us off, welcome to all our panelists. Really glad to have you all on board to this discussion. I'd love to just go around the virtual room and get each of you to briefly introduce yourselves, specifically your unique perspective on this topic and any initial thoughts before we dive in. Judy, do you want to start us off?
Thank you. I'm Judy Chow, the president and CEO of UltraBio, which is an innovative company focused on novel therapeutics for immunological diseases. So while I'm running an organization from biology side and also the technology manufacturing side, I actually starting my career in the tech ops or tech development side, and then gradually grow my responsibility and also get into the research commercial. So very happy to share some experience, particularly also the one before I took on my current role. I used to run the entire global biotech unit for Bayer Pharmaceutical. So happy to also share a little bit from small company and large company experiences.
Great, thanks, Judy. Adam, do you want to go next?
Yes, thanks. Hi, I'm Adam Adler. I work for Gigagen, which is a subsidiary of Grifols. I'm vice president of R&D at Grifols and head of R&D at Gigagen. So we're an antibody therapeutics company. We utilize a proprietary single-cell platform for discovering and developing recombinant antibodies. We do develop traditional monoclonals, but we also created a new therapeutic modality, which is recombinant polyclonal therapies. These are composed of hundreds or thousands of unique antibodies, all that are created from a single cell bank, which, as you can imagine, would require a lot of advances to the manufacturing side I'm excited to talk about today. We currently have 2 assets in phase 1 clinical trials, a monoclonal in oncology and a 1,000 antibody diverse recombinant polyclonal targeting hepatitis B virus for a potential functional cure. And we also have a large pipeline of discovery programs for autoimmunity and infectious diseases.
Great. Thanks, Adam. Elizabeth?
Hi, Elizabeth Dykes. I'm a partner with McKinsey based in Washington, D.C. I've spent about 20 years in life science operations, most of that time at McKinsey, but a short period of time at Merck prior to that. And I'd say I serve life science companies, big and small, across the development to manufacturing continuum, and so can, I think, speak to what we see happening across the industry, both on the innovative side and then CDMOs.
Great, thanks, Elizabeth. And David?
Yes, I'm David Shen, founder and CEO of Propeller Bio. Propeller Bio is just a new startup. We started in last month. But previously, I was founder and CEO of biologics. It's a biotech company. We specialize in multi-specific antibody approach for the immunology and inflammatory diseases. The company was started in 2021, and then in less than 3 years was acquired by J&J for $850 million plus milestone payments. And we certainly bring a very unique approach. So we use multi-specific antibodies, and it will be an interesting topic for today's discussion as well. How do we meet those kind of challenges with very complicated antibody structures? And the previous, I was at the NGM as a senior VP. We brought in about 8 molecules of monoclonal antibodies into the clinical stage. And prior to that, I was a global VP at the Catalent pharmaceutical, and we have several biosimilar molecules approved in US and European market. Thank you.
Okay, great. Thanks, David. And congratulations on that deal. It's very, very impressive after 3 years. So thanks everyone for those intros. I really love the unique backgrounds and, the differing backgrounds that you all have. And we'll bring to this discussion. But to start us off for the first question, I'd love to talk about, novel modalities. Protein therapies are getting increasingly sophisticated and complex. What are the unique challenges around manufacturing therapies like bispecifics, multispecific antibodies, and novel protein modalities compared to the traditional monoclonals? Adam, maybe you want to start this one.
Sure. Yeah. So as I mentioned in my brief bio, we created a new recombinant biologics therapeutic modality. So these are not kind of antibody cocktails. These are composed of hundreds or thousands of antibodies from one cell bank, one cell vial. And we didn't really want to invent the wheel for all the steps of the process. So we actually wanted to take advantage of the existing manufacturing infrastructure that's out there in the world. So we tried to model our manufacturing process as similar to a monoclonal as possible, and then really only innovate where necessary for what was unique to our molecule and our process. For example, we actually, because we have such diverse antibody sequences, we need to make sure each cell produces only one antibody. So we use a single integration site cell line. This ensures the proper paired expression of each antibody. And then we had to figure out ways to develop processes to ensure that this stable cell bank remains very stable, very consistent in its production. The product needs to remain high quality and reproducible, and also with high production titers. And so we also had to work through those processes, and then we had to create some new techniques also on the purification side and the analytical side to ensure we are properly analyzing these products.
And David, I'm interested to know what your experience was, especially, with your experience with proteologics, which were bispecifics, right?
Yeah. So for bispecifics, certainly it has a lot more challenging than the monoclonal antibodies. As I monoclonal antibodies before, and the process is pretty predictable. But for bispecifics, depend on what kind of format or structure of your bispecifics. For example, monovalent bispecifics, the structure just like a regular IgG, but one arm for antigen 1, the second arm for another antigens. So those kind of structures require 4 chains transfection. So you have 2 different heavy chains and 2 different light chains. So just based on that, they make the transfection and the expression a lot more challenging. And you have to make sure there's no chain shufflings going on. Your heavy chain 1, light chain 1 need to be paired specifically versus heavy chain 2 with light chain 2. Most of the time you will encounter scramblings for those kind of transfections. And then the other kind of bispecific is a more bivalent bispecific. So in that case, you just have a regular antibody, maybe the C-terminal, put a single chain free fused to that. So that one actually is relatively easy for transfection because still 2 chains. You have one heavy chain fused to the single chain free, and then the light chains. So really there's no chain scrambling issues, but because of single chain freeze, you have aggregations and a lot of complex clippings issues going on. So you have to deal with that as well. And also, that one also have a potential like immunogenicity issue in the clinical development that you need to deal with, make sure the structure really stable for that. And Stability is the major issue, like Adam mentioned, that to make sure that those clones are very stable, especially with 4 chains, make sure that you don't have a deletions or mutations going on. And then for those 4 chains, the title can be an issue. Sometimes you don't, like a monoclonal antibody nowadays, 5 grams, 7 grams, very easy to achieve. Or by specifics, that can be challenging. Purification certainly is a major challenging in the process. Make sure that you don't purify those chains that have a scramblings or cripplings. And then the other major challenge is really the analytical characterizations. Make sure the final product is your molecule that you wanted. So all these kind of things are quite challenging. And especially for immunology disease, because it's a chronic disease, we need to ensure it's a sub-Q formulation, not like oncology, you can do IV. IV is very easy for formulation, but sub-Q, we have to achieve very high concentrations, like a minimum 150 to 200 milligram per ml range. So in that case, for bispecific, that's another very challenging aspect to make sure you don't have viscosity issues. You can formulate a high concentration and still be very stable. So those are a lot of challenges we have to face.
Okay. And also, you know, tech transfer is also one of the most difficult transitions in protein therapy manufacturing. Curious to know, like, the common hurdles faced there and how companies ensure a smooth transfer from development also to large scale production. Judy, do you have any thoughts on this, specifically because you have that experience, I guess, with UltraBio, but also in your experience at other organizations?
Thank you for that question. So for tech transfer, personal opinion, like many things, when you got the technology from one place to the other, the communication definitely is very important. That's easy to say about communication is important, but that also means 2 different organizations have a different way to do things. We need to align. So in my opinion, the first thing is really to align about the expectation and the fit for the facility. And second thing, a little bit technical, is about, I do think this is a little bit different nowadays. In the past, we talking about tech transfer, most of the time it's either transfer to a facility and also like Sarani, you just mentioned about the scale up. But now I think it's more and more widely accepted as this whole scale out, in a sense, like trying to define that base unit and trying to do more of that. So reduce the tech transfer hurdle a little bit. But I think this is more or less all different organizations need to think through about their strategy on the technical side. So while scale up, it's more economical. But the 2 things to consider, one is then the whole tech transfer hurdle of a lot of things that all need to take into consideration, the chance of success and the time it takes for scaling up. Second thing is also the demand, right? Because we always have this forecast and demand, and sometimes scale up at the end, maybe the supply and the need may not align very well. So in sum, I would say, in general, in the past, there's a huge about the scale up challenge, but I personally feel like then probably worse for the organization to really thinking about, do you really want to scale up or may as well just cut the time by scale out, meaning it just based on those units, see the demand for a while until it's really stable commercial supply. So I would say communication to overcome all those connection, make sure all different organizations can talk on the same page. And second thing is really the overall technical and business strategies.
Great. And Elizabeth, I'm curious to know your experience with that, you know, just working with a lot of clients, you know, the tech transfer process, but also, you know, talking about manufacturing facilities, you know, the thought process behind choosing between single-use bioreactors and versus stainless steel systems. What's your experience there?
Right. I fully agree with what Judy just shared. And I think if we think about like your facilities, what we see in general, and perhaps not surprising, is that like your capital choice and the facilities that you build have like the biggest impact on the ultimate cost of your product, which again, not a surprise because you have that fixed capital base there, but I think it makes that decision-making even more challenging so early on when there are a lot of unknowns. And I think that, you know, Sarani, to your point, plays into what we see on single-use versus stainless steel choices. I think really it comes down to a few decision points. I would say cost, agility, and then of course, like technical fit, you know, compatibility, perhaps to spend a bit more time on the cost and the agility. I think, you know, no surprise, you're effectively trading off like OpEx costs in like higher raw material cost for when you have single use versus a higher CapEx cost for stainless steel. I think what we see is that stainless steel really pays off when you are then able to fully utilize that facility, which means that it's more applicable or more advantageous when you have, you know, a blockbuster drug, very large volumes, and so you're able to process larger batch sizes through that equipment. Or if your, I'd say, pipeline strategy, which is a bit more challenging, means that you will develop products, say, at that similar batch size. So you're able to have, say, like a multi-product facility and get utilization that way. I think what Judy just said is like more what we're starting to see in some of the newer products, which is, you know, given a multitude of factors like probability of success, both in general for a particular molecule or asset, but also in terms of like commercial success. We're seeing more and more companies stay at, say, like a 2,000 liter scale or something like that, which is more advantageous for single use. And then that allows you to more scale out as opposed to scale up in bioreactor size, which cuts the development time, but then also like risk manages any like capital investment that you may have to make. And then, you know, if needed, and obviously if the ROI is there, you can come back kind of post-approval to develop a larger batch size, but you at least get to market and can test the commercials out as you see.
And David, I see you nodding a lot there. Do you have any thoughts on you know, Elizabeth's points?
Yeah, certainly. Like in terms of scale up, like I think the facility fit is so important because we had a challenge when I was at Tavaton. We moved the manufacturing from one facility to the other. And just because the centrifugation systems are different, it takes a long time to figure out how to really transfer the process from one facility to the other. So that part, I think people really need to pay attention, make sure the facility fit is so important for transferring the manufacturing. And then the single use versus stainless steel, I totally agree with Elizabeth's comments. Whether you want to invest early, the big capital, or just do single use because it's a low investment, very quick, especially for biotech company. The single use is really, really useful now. Timeline is short and investment is little for that, and it's very flexible.
Adam, do you have anything to add on that in terms of all those points, tech transfer and also the use of the facilities?
Yeah, a more general approach in terms of finding manufacturers, make sure the tech transfer process is going to be optimal, is that whether you're trying to manufacturing something novel or in complex or something traditional and well-known, we find it's really critical to take the extra effort to find the right manufacturer where they see you as a partner and not just a customer. So we find that, especially if you're trying to put that manufacturer a bit out of their comfort zone, you want to be able to work really closely with them. You want them to be very flexible, and that for each step of that tech transfer process, they're working with you, and that will hopefully lead to better success at those later stages of the scale up. And so this is useful for any stage of company, but especially if you're virtual, you're only relying on external partners for everything. Having that close relationship, making it feel like you're part of that team is going to help lead to that success.
Yeah, and I guess, speed to clinic is often a big focus for, biotechs and, the followers of these biotechs, namely investors. But what strategies can biotech companies adopt to accelerate development and manufacturing timelines without compromising quality or regulatory compliance? If anyone wants to jump in, Judy?
No, maybe I have a little bit interesting background. As I mentioned, I start my career on more in the industry, like more tech development and tech up. So often this is probably in this panel, everybody understand, and maybe so as most of the audience will understand about on this side, the most important thing, you don't get yourself on the critical path. Meaning the project wouldn't be slowed down because anything, the development or the manufacturing. So you got to be just the clinical. So in a sense, also on the flip side, that means that nobody kind of care about it, right? Just develop and deliver the needed material and doesn't really want you to spend too much time on that, while there's many issues as we just discussed a moment ago, because now all the antibodies or protein are development the same. And so, but on the other side, I must say that my current job as a CEO on a novel therapeutic development is very focused on the timeline on the clinical. I kind of joined the dark side about surplus people about don't even talk about this. So let's just push that to phase 3. So managing these two sides, I think it's hard to say this one answer to see how it goes. But I think jointly we all agree the speed to market, the speed for development is still the priority. Meaning to show the clinical efficacy is the first, but the difficulty definitely on the tech development, tech ops side is then one, when it's needed, and maybe you need to fix the cell line and the cell culture processes, many things need to change and become like out of time. And then also, I think it's really wonderful about the particular FDA have this accelerated approval path. And that pretty much means once the proof of concept, the biology, the efficacy is there, quickly you just need to turn to phase 3 production. That is really the very high challenge. And if I have any wisdom to share, I think really from the tech development, tech ops, I need to stage this and then stage this to make sure it's still not on the critical path and having some prep work upfront investment. And second thing, I would definitely think like raise that red flag about, well, make sure it align with the whole team. Yes, we can take the risk not to change the cell line or something like a fundamental require a lot of a long lead time, but everybody's fully aware of that because otherwise at the end, this is going to be on the critical path. It's not pretty. And I think as we all work so hard for patients and the patient will be waiting for the drug, that's the least thing we want to do. So I think definitely strategize the whole plan, but also to align with the team, the development timeline to make sure this is all the risks everybody all committed to. Later on, then this may actually take more time.
Yeah. And Elizabeth, I see you nodding a lot there. I'm sure you have a lot of experience with your different clients on this.
Yes, I fully agree with what Judy just shared. Maybe just to share like a couple of data points. I think this is probably one of the top discussion topics over the last, I'm going to say probably 2 years. It's probably a bit before that, but it seems like every company is now trying to look at particularly the CMC development timelines and exactly to Judy's point of like, how do we stay off of critical path? Like, and I think people have found that they can do it on a single asset and can, put more resources on, that's like the typical lever. But now the question is, how do we systematically actually move our whole pipeline to patients faster? I think we actually see leaders starting to set ambitions around like 1,000 days, I think is the latest I've seen from like DNA to launch, which is quite different than the typical timelines. And maybe just to share like a few different levers that companies are starting to pull, and again, always important to balance this with like the right risk taking and the right like quality and compliance of the development first and foremost. But what we see, you know, I think to Judy's point, like a look at more of a 2-phase development approach as opposed to 3-phase for exactly that point around accelerated timelines. You know, I think oncology products are largely already there. And this is really about what we see is people mapping out effectively their end-to-end development timelines and what decisions are made where, and like therefore where collectively are we ready and able to take risk. There's a bit around agile like resource allocation as well across products versus having like set standard teams. And then obviously like digital playing like a pretty big role in like in silico modeling, automated labs, digitizing workflows, things like that. And then I think very lastly, depending on obviously the stage of company that you are, we see people really building like playbooks, if you will, that standardize like these decisions and parallelization activities and how to make risk decisions so that it's not individual teams making them, which tends to be less like effective and efficient overall. And more like a standard approach that like not just CMC, but also quality, R&D, you know, other colleagues are all on the same page, again, about the process and the risks being taken.
And you mentioned, you know, the digitization. So I guess that brings me to like the next sort of area of conversation, which is technology and innovation. So I'm curious, you know, on to our panelists, how digitization, automation, and AI is influencing decision-making in the manufacturing, you know, of complex protein therapies, whether it be, you know, early stage development with things like cell line selection, process optimization, to the latest phase manufacturing scale up. Perhaps some of you can talk through your personal experiences. Adam, maybe you have some experiences with, you know, the early stage use of some of these novel approaches.
Yeah, the use of AI, machine learning, it's really exploded over the last several years, especially as it relates to antibody therapeutics. So Gigagen's actually been utilizing these techniques internally for some time now. Most of those efforts are really focusing on new models and techniques for identifying and characterizing antibody sequences. These are for our early stage discovery programs, help us identify issues that might be coming up early, or helping us select the right clones for our polyclonals. And so it's early, but it's really starting to grow. And then another area where we're starting to look into utilizing, again, AI and machine learning type techniques is for optimizing bioreactor conditions. So it's really critical to have high levels of production titers, but still maintain that same high quality product. For us, it's a little tricky with our polyclonals because we are limited to a single integration site per cell. You can't just throw 1,000 copies into a cell and get the highest titer possible. So optimizing, finding other ways to optimize production levels outside of the cell line development itself is something we're now looking into, and there's a lot of data out there. So utilizing AI tools and models, we're hoping to really kind of revolutionize that step of things.
Really interesting. And Judy, I guess you have a lot of experience with, you know, the industry and its, you know, move into pharma 4.0. Can you share your thoughts on, you know, the whole AI digitization part of it, especially, you know, going on to the development scale up?
Thank you. So if I may, I'd like to go back to the AI, just add one more point there and echo what Adam just said for the development, help to optimize and also accelerate the process. That's very impactful. And the other thing, I definitely, in my former organization, like large manufacturing and all different sites, we have been using in the past already, using AI as part of the logistics, also the resource management. And I think as Elizabeth mentioned, how you build a facility and run your facility, frank speaking, in some sense, weight even more than the cost of goods than the process. I really hate to say that because as a technical person, we like to believe we change the cell line tighter 2 times and make a 2 times impact. And sadly to say, that's not the case. Really is the whole manufacturing, how you run that. And what do I mean about AI play important role? So in my former organization, we have this program. You can even tell, like, then if you have one operator not be able to come in, not just the whole the human resources side, but also the logistics about how the impact and the raw material and how much it slowed down. And those really using the power of AI will be much faster to react to it than the human intellectuals are trying to figure it out. So back to the question about the industry 4.0. In my opinion, I think AI is a little bit beyond the 4.0, and 4.0 is more or less really the Internet of Things and big data. So then AI is later on beyond 4.0. But let's focus on the 4.0 first. I think that itself, I see the biggest benefit now as I'm running organization more about let's just call it the dark side about product, and I don't care about the CMC or something like that. And the most important thing is really this loop system. I've been saying this a lot about. Basically, the whole internet and the big data should be a platform to start with patients' data, and then really having this deep analysis about the supply, the need, and modification. That will give the manufacturing, so as the process development, the needed info about how much improvement really need to be done, how much the production, the productivity, and what kind of facility fit will be better. And also so as the logistic about coming back, the deliver to the patient. I see this is a well-connected circle all the way, not just manufacturing, but also from the patient side, the clinical side, the hospital. And that will be ultimately to be able to bring down the overall cost. So if we can really working towards a 4.0 stage, the big data analysis, truly will make a huge impact for this industry. Otherwise, we'll always do this a little bit art about, well, you produce this and you have best hope about the need, and maybe this is not what patient need. As we also talking about the formulation platform, is that sub-Q, is that the pre-filled syringe, or many things, all those costs, all should be managed by connecting the patient, the hospital, the clinical, manufacturing, and then with this whole loopback system. I think that's a spirit about 4.0.
And Elizabeth, you have more of a bird's eye view on this. Are you seeing, measurable impact across different clients, moving more into pharma 4.0 practices?
I think the short answer is like, yes, but. So I think there are definitely some front leaders in the biopharma space that are seeing impact. I think we've heard some of the examples here today, particularly I'd say tighter, like maximizing titers from the cell lines, maximizing yield in the manufacturing sites. We have also seen, I think, real impact from like advanced analytics enabled like root cause problem solving. You can think particularly about like human error deviations, which is like usually the largest group in a site, and like, let's be real, no one really knows what the root causes are, but they recur. So the use of some of the industry 4.0 tools to understand, for example, like better shift structures, et cetera. I think Judy also highlighted a few like agile, like resource loading. We see that in particular in like QC labs, like what's the next best analyst to test the next method to actually release the batch. I think those have gotten like some pretty good traction. I think there's some early leaders now in the Gen AI space with like human in the loop on like actually supporting root cause investigation or report writing out of non-conformances, like technical report authoring, data, like initial pass, like data analysis or trending, whether that's in the development space, like regulatory submissions, annual product quality reviews. I think we can all think of many places where there's a lot of people like analyzing data and writing a report, like that can largely now have a first pass taken. I think if we're honest, though, the life sciences industry is quite behind other industries. So there's a lot to learn and be inspired by, particularly like CPG companies, even some like automotive or like industrial goods companies that are really, I think, Judy, to your vision, like really truly connecting customer and patient all the way back through their supply chains. I think pharma is quite behind that at the moment. So I look forward to that being one of the big shifts that happens over the next decade.
Great. And so I wanted to talk a little bit about also regulatory considerations because, you know, within the current environment, you know, political environment as well, you know, things are changing, it's a shifting dynamic. So regulatory engagement is often critical for biologics manufacturing. How do companies successfully work with agencies, anticipate compliance challenges, and align manufacturing processes with, you know, the evolving regulations? Adam, perhaps you might like to give us the perspective from an early stage, having discussions, you know, earlier in the pipeline.
Yeah, we had a unique challenge of introducing a new modality to the FDA. And we found the way we ended up doing this was through multiple rounds of interactions with the FDA. It was spread out over multiple years as we were figuring things out ourselves, how to manufacture, how to analyze them. We focused a lot of time on what was unique about the recombinant polyclonals versus, say, traditional monoclonals that they may be familiar with, or actually plasma-derived polyclonals. It's actually more similar to what we're doing. And what we found is that we'd learn lessons from every interaction. We might have a goal that we want, or they may have a goal that they want. We take those lessons learned, show in subsequent interactions that we listened to what they said, build upon that. The agency then became familiar with us, familiar with the product. They became comfortable with it being a novel product. And when we got to the point of actually doing IND submissions, it was very successful. It was a lot of the same people in that room, which was great. And it was not nothing was a surprise to them at that point.
Anyone else have any thoughts on regulatory discussions that might be, you know, shifting in their, you know, I don't know, their form of discussion?
Yeah, I will be happy to. So especially for those innovative biologics, we mentioned before, speed to clinical is so critical, and then certainly regulatory is one of the key aspects to make sure that you can really get into clinical as quick as possible, like pre-IND meetings. Like for us, in order to really speed up on the CMC side, we usually use pool for tox. We're not using single clone for tox. Pool for tox really cut down a lot of CMC timelines. But you really need to talk to regulatory to make sure the regulatory is comfortable to using the a mixed clones or pools for the tox study. At the real phase 1 clinical trial, you use single clone, and then you have to do a lot of comparabilities, make sure that the pool qualities are very similar to your final clones. So those kind of interactions are very critical with FDA, and then early informing them your process really will make things very smooth at the R&D stage.
Great, thanks, David.
And I will add one, maybe a little bit controversial point here. There's no doubt we need to go digital, collecting more information on the biomanufacturing. And so then we know, I mean, in some sense, we used to say, right, the process is the product. So then now we should be able to characterize all the process, getting all the information. This is nothing to do with currently the regulatory landscape change, but given if the industry has so much data, and I think it's no doubt the regulatory, I haven't heard that, but I think it's probably coming about then we'll ask for that level of transparency. And so that will become really making the industry very much exposed. So on one side, for the trending, for the direction we are going, there's no doubt we should collect a lot of information. But on the other side, I think this will need to have some level of alignment. The regulatory probably demand like the in-process even more about to demonstrate, really have the consistency. Something we jointly together need to work with the regulatory agency to make sure those information truly bring the value and not slow down things down, but at the same time really become very informative knowledge for us to make decisions.
Mm-hmm. Elizabeth, anything to add there? How you advise clients, and I'm curious, how early biotech should be engaging regulators or looking to engage regulators on manufacturing.
Yeah, I don't know if I have a specific answer to that question, but I think like what I would say is there is, like, to Judy's point and I think Adam, your point, on whether it's digital innovation or like new modality, I think like the FDA, like at the end of the day, like wants to also innovate. And so like the early engagement and like the collaboration, although I think sometimes can like feel scary or different. I hear that from some clients. Like that is actually the most helpful thing to do. I think the FDA is also experimenting with digital and AI. Like to your point, Judy, like I don't think we're that far away from a time when an inspection is download us your like give us your data. We are going to like have AI analyze it, and they're going to start showing up with deeper insights than what you yourself may have. And so I think, again, I think, Sarani, to answer your question, like leaning forward and like proactively engaging in either like roundtable discussions or the one-on-one shaping to bring them into your problem solving, whether that's a new modality or a new way of working, I think is only beneficial.
Okay, great. And I'd love to just finish this discussion off with looking at, CDMO strategies, relationships with CDMOs, and the overall industry outlook with these types of partnerships. What are some key considerations for biotechs to think about when choosing a CDMO, like key differentiators, technical capabilities versus cost, or other considerations like flexibility and partnership with the CDMO? David, perhaps you might like to start off and give us a little bit of your experience being the founder of Proteologics and having to navigate those partnerships.
Yeah, so like for small company like us, the speed of clinical certainly is very critical when we choose CDMOs. So basically we use three criterias: timeline to clinic, how fast they can do, the quality, and then the cost is also very, very sensitive to us. But sometimes quality and the cost is going the different ways. If you have a high quality, certainly the costs are going up. So we have to think how balance are these factors. And then when we choose CDMO, really we want a one-stop shop type of CDMO. So that means once we give them the DNA sequence, they can just take from DNA to cell line to process development, GMP manufacturing, fill finish, even packaging, labeling. They can do everything in one-stop shop. So that's certainly give you a lot of advantage in terms of speed. And then the other aspects we're really looking for is do they have a platform technologies so that they don't have to change the process too much. So that's another way to ensure the speed to clinic. Like for bispecific, especially whether they have experience with monovalent bispecific or bivalent bispecific, those encounter different kind of challenges during the manufacturing. And then the analytical capability is a key for all these complex biologics. So we put a lot of focus on their capabilities on that. And the other aspect we're really looking for is really, do they have all the regulatory filing document templates available? So in that case, we just do fill in the specific informations in the documentation so that we can get to the filing stage very quickly. So all these factors putting together when we choose, and then certainly the slot availability, the flexibilities, and especially your relationship with the leadership teams of the CDMOs are very critical to us.
Okay, great. Thanks, David. That's really insightful. Adam, what about your thoughts on what's going on with Gigagen?
I could kind of bounce off of David's last point about slot availability. So we do find there's often a battle for bioreactor space. And this became really apparent during the COVID pandemic. And at that time, we were a small independent biotech. We weren't a Grifols subsidiary at the time. We were making an anti-SARS-CoV-2 recombinant polyclonal, and no big CDMOs would help us. They actually had never done a GMP manufacturing run at that time. And so what we ended up doing was we found a lot of smaller CDMOs that actually fit the criteria I talked about earlier, which is really being open to flexibility and being okay with a unique process and having to learn something new and working closely with us. And we actually did something that's maybe atypical, which was we selected 2 of them, actually worked in parallel because this was a completely new process. And we didn't really know for sure how well it was going to go. And so we just wanted to kind of have that backup, able to find 2 groups willing to kind of do this. And then it went well, really well, I'd say. We ended up leveraging the knowledge that was gained from those processes. And now we're applying it to all the different groups that we're working with, including larger CDMOs. And it's helped us learn some, I'd say, some useful tips in a sense. So having people be on site, person in plant, as people might have heard, is really useful, especially if just to make sure everything's going well, no problems are coming up. And while obviously not always possible, having a local manufacturer is even ideal. You could drive into work, go to the CDMO that same day, versus having to say fly someone around the world to keep that close eye. But yeah.
Okay, great. Thanks, Adam. And Judy, what are your thoughts there on partnerships?
Yeah, building further on the point David started, I think Adam elaborated very well. I think the CDMO business, it sounds like it's about technical capability and also experience, but really at the end of the day, it's a relationship business. And again, I won't repeat what the point David and Adam already say, but I would probably adding one more dimension about nowadays, today's environment, is the funding for many biotech company is very, very limited. This is really bad. So the biotech is in a very miserable situation about the funding is not coming in. So all the biotech company are fighting, trying to get the way to survive. But at the same time, also the huge pressure to our industry is about this pricing cut and everything. So something needs to change, right? And so then most of the biotech companies still pay exactly the same price to CDMO and CRO. Something's wrong. So I feel like I'm not running a CDMO organization, so maybe a little bit easy for me to say, but I would say if I will run one, I will have to have the self-discipline, thinking about something I need to offer more than, I mean, I can still keep my costs to my clients, but I need to offer more. And I have to say then being on the client side for us, and without naming our CDMO, I think that's what our CDMO partners been doing. They don't reduce their costs, but they do a lot of flexibility to make sure it works for you. Because otherwise, those really, I think the big issue is the innovation will stop. And this is really bad for the whole society, for patients, for many things, right? So this is not about we need to cut CDMO, but more about the way to build a relationship. It's not just about, doing build this whole partnership spirit, but also realistically offer some flexibility on the payment and things like that. For me, that's what I'm looking for. Like then if they can see the flexibility is needed. And again, I don't want to overbrag, sound like Ultrabio have tons of money. We are the company, luckily, and I really do think it's luckily last year we raised the biggest Series B round in the past 4 years. So we have that luxury, however, still very matters about having a good partner, understanding the cost sensitivity and the speed to market, and then create all those possible terms to build that relationship in that way, in the business term. I think that's very important today, particularly.
Super insightful. Thanks, Judy. Elizabeth, any final thoughts on that with your sort of bird's eye view and, how you sort of, I guess, advise clients on how they should be forming partnerships?
Yeah, I won't repeat what has been said. Maybe like two points I would add. One is it can be really tough also as particularly a biotech, like you are competing for the same capacity oftentimes with, the likes of like Lilly and Merck and AstraZeneca. And, to your point, Judy, like deeper pockets, oftentimes larger, more sustainable volumes. And like for a CDMO business model, like that is great, right? You want to fill your sites. So I think that implies like two things that like I think everyone has raised here. One is as a biotech, like who are you targeting and how do you then like partner with them? And we've seen some examples of like longer term partnerships so that it's not like a single asset or like a couple of runs, but could you partner with somebody on your portfolio, which then, again, while there's still uncertainty in that, that's like a much different like negotiation bag, if you will, than like one single product. I think the other is you also will like only be able to partner with who like you can, that not everyone's going to partner with like top tier CDMOs. And so if you find yourself, not with top tiers, how do you like actually grow together? Right. I think, Adam, those are some of the examples you gave, like Judy and David as well. And then I think what we see is when it is a little bit more of like skin in the game from the biotech side or that partnership, you're then more apt to like jointly be flexible on like multiple terms, like Judy mentioned. And so just, again, more nuanced than that, but a few strategies to think about, like increasing your chances of having a good partnership.
Add on just a little bit about for any audience here from biotech, and I used to run big organizations, and that's the same thing also, like Elizabeth just mentioned about, even for big organization working with CDMO, and need to strike this kind of contract about commitment to the whole entire pipeline in order to be able to get a good contract. So it's just small company alone. For big pharma also, that's the way to have this business relationship to build a success.
Yeah, totally agree with Judy's comment. Because like for a small company, in order to get a lot of attention from the CDMO, you're competing with a big company, Merck, whatever. So certainly you don't have the capacity to compete. So what we use is we said we build a strategic relationship. We commit a whole pipeline, like we have 7, 8 molecules to commit with you. In return, we get a lot of attention from you. So that is something we do quite frequent, and that's the way to get attention.
All right, great. Well, a lot of insightful discussion there. Let's move into the Q&A portion. We do have a few minutes maybe to take a couple of questions. I do see some that have come in. So the first one is, thanks for pointing out the importance of scale limitations. With respect to polyclonal manufacturing from a single cell bank, is scale-up even more limiting to ensure consistency in cell titer, protein titer, viability, glycolization, etc.?
Yeah, so it's an interesting question. And I'd say, no, it's not more limiting if you are prepared. And so we've been told for years that what we're trying to do is just not possible because of the diversity of the cell bank, doing it all at once and scale up. So we put a lot of effort into solving all these problems, essentially proving the field wrong. And one thing that was really critical is just figuring out the processes at the small scale first and do it in a way that is scalable. And so, for example, having the same brand of bioreactors in our labs here in California that the large manufacturers are using, we figure out those processes here, we tinker, we modify, then we do a great tech transfer process, they're mimicking it, and then it scales. And so obviously I can't go into all the details of how we figured out these things, but we are able to scale and we're not limited, even though we are doing something a bit more complex.
Okay, great. Thanks, Adam. Let's move on to another question. This is a question for Judy. What factors determine whether to restart a cell line? Would it be mainly economic considerations, which would override the critical path impact? Would it be okay to accept being second to market but have a superior product?
Yeah, thank you for the question. So there's no one size fits all, unfortunately, and also unfortunately, it's never been okay to be second to the market because you're selling. So that's the reality. And however, I think it's really based on the product itself, like the market projection. Let me share with you my personal experience. This is actually, of course, way back. So I was the one leading this Embro new cell line after Embro already a blockbuster drug, and why it need to be changed. It's a serum containing process and many things. This is a post-market improvement. And for now and going into the market, if you happen to have PERDA, changing the cell line will actually slow down for going to the market. Like I say, it's never been any case will be that's okay to take it back. However, this is something about if it's any special niche for the product itself, if you're not competing exactly the same target, the same, if like back then about the anti-PD-1, for example, when Merck and BMS competed, I think there's no way you will have which cell line is better, you go back, but you will have to come up with a plan. So sorry, there's no one straight answer, but really factor in the product nature and also the market projection. And most of the time, indeed, it will be the post-market change. However, let me just not confuse the audience, because definitely it's very common people make the change before entering phase 3. So that basically having some level of proof of concept in phase 2, and then that is a golden spot, I think, for most of company nowadays. Always use that.
Okay, thanks, Judy. Perhaps time for one last question. For timeline acceleration, how much is that a consideration for newer modalities other than monoclonal antibodies? In other words, is time still the most important factor as opposed to titer and quality, etc.? If anyone wants to take that.
I would say quality will always be upfront, right? Because then if you don't have the quality of the product, and then there's no way, because the whole BLA approval process will take long, so that doesn't pay off. But definitely then second to that, it's still, time is still very important, and open to other panelists' answer.
Because I would say it's a balance across all of it. You want to be fast, you want to have high titer, but you also have to have high quality. And so all those considerations have to come into play. If it's novel, it means you might have to spend a little more time, maybe for your first version of it, but then maybe the next iteration of that novel product, you've learned a lot of lessons, you can speed up that timeline and still have those high titers and remain, keep that high quality.
Yeah, really the balance, like for the new modality or novel pathways, you want to get into human as quick as possible to demonstrate whether your molecules have a clinical efficacy or not. So in that case, you don't want to spend too much time in the process part and then demonstrate your molecule not really working in human, right? So you need to balance that. So we try to balance those ones very carefully. Speed is certainly critical to demonstrate if the molecule is working or not. Once you have a validation, clinical validation, then you spend a lot more time trying to make sure you have a robust process, qualities to make sure you can really scale up for the phase 3. So the balance of these two aspects are very critical.
All right, well, we have hit the hour, so that's all the time we have for this session. Thanks to all our panelists for their very dynamic discussion and expertise. Thanks to the audience for joining and for the questions, and thanks to Catalent for sponsoring this discussion. Just a reminder, a recording of this webinar will be available on demand tomorrow to rewatch or to share with colleagues. But for now, I'm Serrani Fernando on behalf of Endpoints News. Thanks for joining us, and we'll see you at another Endpoints webinar.